Hi, this is Miss Litton, and this is a review session. Say hi. For Chapter 20, Viruses, Bacteria, and Archaea. So let's hit all the salient points. The first thing I would probably review, if I were you, is to review virus or differences between viruses and living cells. And so you want to be comfortable with that. So viruses are non-cellular. They're not alive. They require a host. They are obligate intracellular parasites. They only reproduce by taking over their host cell's machinery. As far as their origin comes from, remember we talked about they could have come before we even had the 3 domains. Is that the one I pulled out? Yes, okay, good. Before we even had the 3 domains. But because they are so host specific, then that also lends credence to the hypothesis that they, because of different ways that DNA can move around, crossing over, what are the jumping genes? Transposomes, that those types, see, they knew that. Go honors. That they could have evolved from their host. The next thing I would definitely know is the structure of a virus. And all viruses have 2 things in common as far as structure goes. What are those 2 things all viruses have in common? They have a protein capsid. What's the second thing? A nucleic acid core. And that nucleic acid core could be DNA, it could be RNA, it could be single-stranded, it could be double-stranded, it could be the sense strand or the anti-sense strand if they were single-stranded, okay? So that they have in common. What's different is they could be packaged with different enzymes, and they may or may not have a what on the outside of them? A membranous what? Envelope. It may or may not have a membranous envelope. Who tends to have membranous envelopes? What kind of viruses? Animal viruses. And they acquire that membranous envelope from their what? Host. From their host. Okay, so that would be the next thing I would for sure be comfortable with. Okay, remember APBio, mister? Turn to somebody next to you and then don't look at the screen if you can hold off, and try to remember the stages of reproduction. Ping pong back and forth or something without even looking. Okay, so the way we remembered it was APBio, mister. And there's some variation on that theme. So attachment, somehow it's adhering to the cell. Penetration would be just getting that nucleic acid in there. But the whole cell could go in as well, right? Bio, there's going to be what does bio stand for? Biosynthesis, making of more viral components. And also any enzymes that they would be packaged with. And then the M is for maturation. That's the point where the virus is compiled into one unit, all of its parts. And then release, in this case, it's a bacteriophage. It's lysing the cell and killing the host cell, or maybe it might bud out in some sort of fashion. So we're just going to see variations on that theme. The next part, some variation on this theme as well, is the difference between elliptic and a lysogenic cycle. So whoever you're sitting next to, verbalize, maybe whoever has the darkest shirt on be elliptic, and the other bio buddy be lysogenic. Go ahead. See if you can verbalize that. Which one would you classify as quick and dirty? Elliptic, yeah. Because in the elliptic cycle, you're going through those APBio, mister stages right away, and you're going to lyse the cell and release and move on to your next cell. If you're in for the long haul, okay, then that is going to be the lysogenic cycle. And the way we remember that is in lysogenic, in genic I hear the word what? Genes. So it's getting in your genes. And so, sorry. And so you still have the attachment, the penetration, but then after that stage, then you go into integration. Okay? And in integration, you're going to integrate into the host chromosome, and you can lay latent. Do you remember what it's called? Hopefully the answer isn't on this board, I can't remember. When you lay latent inside the host cell chromosome, you're called a something. Prophage. Yeah, prophage or prophage. And so you're like a professional virus, and you're inside the host cell. And then every time that host cell replicates, then the viral nucleic acid is getting replicated as well. Now, when they leave the lysogenic cycle, they may leave with a little bit of the host cell's chromosome and carry that with them to the next host they go and invade. What do you call that term where you carry viral nucleic acid from one host to another? Use a virus as a vector. Any guesses? Yeah, you take DNA from host 1, the virus takes DNA from host 1 and brings it and integrates it into host 2. That is called transduction. Say that word, transduction. And we talked about that underneath the umbrella of what are the 3 ways that you can get genetic recombination in bacteria. One way was transduction, where a virus brings you new traits. One we've already talked about, if I say Hershey and Chase, you should be thinking about transformation. And that's where you pick up nucleic acid from your environment, which then changes your, oh, not Hershey and Chase. That's why you're looking at me with big eyes? Because what should I be thinking about? Griffith, right? Where the mice picked up the S strain and the R strain. You're like, yeah, that's why we didn't know. Transformation is when DNA picks up, the bacteria picks up DNA from its environment. And then what was the third way? It starts with a C. Good Lord. Conjugation, and that's when you would have to use your sex, starts with a P? Pillis. You got that one down. All right, so I stepped away from viruses for a minute because I could connect talking about how viruses can pick up their host DNA if they are a virus in the lysogenic cycle as a prophage, and pick up a little bit of the host DNA and carry it with them to their next host. And that's called transduction. Okay. All right. So then we saw variations still on this theme. We talked about animal viruses. And because animal viruses have a membranous envelope, then they have the ability, they know the secret knock, they have the right spikes that get them invited into a host. Okay, and when we talk about the immune system, we'll talk about ways that can be combated. Okay, some antiviral techniques that cells have. So they get brought into the host. Their membrane on the outside has to get removed. Their capsid has to get removed so you can get at the nucleic acid and then generate more and do biosynthesis and make any proteins that are also needed. And then when they leave the host, they may or may not destroy the host cell. They may lyse it and you would get a sore, like a cold sore, or it may bud out and just leave that cell and then go to infect another cell. And then the other variation on the theme we saw was a retrovirus. And why doesn't the lightest shirt person explain what they remember about a retrovirus and how it functions and why it functions that way? Go ahead.
Steve, do you know?
Andrew Nevon, please come to the office. Andrew Nevon, please come to the office.
Okay, so retrovirus gets brought into the host cell. It does have a membranous envelope, and in this case, and it has, what kind of virus is it? A DNA virus or an RNA virus? RNA virus. Single-stranded or double-stranded? Single-stranded. So the first thing it needs is packaged with it in the process of maturation, and it directs the synthesis of this during biosynthesis. Packaged with it, it has an enzyme called yes, which can then make a DNA copy of its RNA, and then it also has another enzyme that's made replicase, which makes it double-stranded, and then it has another enzyme integrase, which gets it into the host cell chromosome. And then what will happen is once it's in that host cell chromosome, it acts just like we talked about before as a provirus or a prophage and can lay latent until something triggers it to leave the host cell chromosome and enter into biosynthesis and do, were you looking for someone?
I have a question.
Okay, I'm recording this for YouTube right now. Is it something that can wait or is it an emergency?
I don't
Okay.
know. So, do we have to do this?
I can't do that right now because I don't have my glasses.
It's the,
The one that, the short one had to be done before.
because I did one of
There's
those.
two different dates on there. You will feel free to pull out the Chromebook and take a look and have the dates on there. Whatever the date is, I have the right date on that.
Okay.
Okay. I'm sorry. And so then something will trigger it to leave, so it's not a provirus anymore. Usually if it's like something like HIV, it's something where the immune system is having to get together a response, and so that can trigger it to move into the biosynthesis stage. Okay. Then the other part we talked about is we talked about these emerging diseases that are increasing in their geographic range, and there's a connection between these emerging diseases and what? What was the connection? Why are we seeing more of them? Could be travel, and what else? Yes?
Temperature?
Yeah, climate change. Climate change. Because then the vectors usually of these diseases are able to live in warmer climates, and as you have areas that are warming up and staying persistently warmer, then those diseases are able to spread more readily. We also talked about a couple other words, pathogenicity and virulence. And if it's a pathogen, it can cause a disease, and how badly it impacts you is the virulence. And we said these are not the only ways. We also talked about viroids, which are little naked pieces of what? Remember? Yes. Good. Little naked pieces of RNA, which can cause diseases, especially with crop plants. You can see the difference here. And then we also talk about Creutzfeldt-Jacobs disease or mad cow disease, and these are abnormal proteins, and they have the tendency that, to exert a lot of peer pressure and get other proteins to also misfold.
Yeah, I need to talk to you about something real quick. But
Okay.
if you're doing a review.
I'm doing a review. Is it real?
It's just like a little talk.
Okay.
I can wait.
Okay, I'm gonna come by. Okay, come back. All right. Very popular this lunch period. Okay, I turned on, I think it's countries. It's 41 number. 41 number. Here, come over here and whisper in my ear while these guys are answering these questions. Yeah, sometimes people fill them out early because of these programs. If you want to do that, you can. It's on non. Yeah, and just answer all the questions. Just hold them Give me specific things that specific things. Then I
So I'll come by
include them.
tomorrow and with the
It's not due till March.
Yeah.
You don't have to come tomorrow.
Okay, got
It's fine. And Thursday I'm out at a conference, and then, but I'll be back on Friday. So it doesn't matter. You give it to me next
Okay,
week.
got it.
doesn't
Thank you.
matter. Okay. No problem, sweetheart. Okay, what did we miss? Somebody picked D. No. How are you gonna remember this one? Yeah. Yeah, a profile, I think professional virus, that's how I remember this one. A professional virus knows how to integrate itself into the host chromosome. Let's just say this one. I don't want to wait for you to type it. What's the answer here? Yeah, APBio, Mr. So attachment, penetration, biosynthesis. And then what's the next two? Yep. Your next class, I believe, normally it's at 1:05, it's at 1:10 today, just so you know. Okay, it's not gonna translate the host DNA, because that's not the job of a virus. A virus doesn't wanna do that. A virus is all about itself, right? It's not gonna do anything for the host. Okay, so, it will transcribe its viral DNA into complementary DNA, which can be integrated into the host chromosome. All right, now, the next part on bacteria. The key things I would probably know about bacteria, I would review those structures that we have already discussed in previous chapters. I would talk about the insides, things like there's no membrane, there's no partitioning that we would see because there's no membrane in closed organelles. You don't have a nucleus, you have a nucleoid region. Look for those differences. You have a single circular chromosome instead of a linear chromosome. So that's the inside. Then you could maybe talk about the order of the barrier, and it's gonna go to the membrane and then a cell wall made out of peptidoglycan, and then you're gonna have some covering potentially on the outside that could be slimy, it could be hard. We talked about gram positive versus gram negative, and gram positive, remember, stained better, so you could see it. I don't think they'll ask you something about that. Then attachments. What are the different attachments that you could have? You could have what? Flagella, you could have a hey. You could have a sex pilus, and you could have what? Fimbric. Good, for attachment. So those are the things I would know about bacteria. You could talk about how it could have an extra piece of DNA, which is not in that picture, which is here, and we call this plasmids, extra rings of DNA. You could talk about its reproduction, which is through binary fission. It does not undergo mitosis, like you would see in a linear chromosome. We talked about ways to have variety. So transformation, when you pick up DNA from your environment, conjugation through sexual, through a sex pilus, and then virus acting to be a vector. Also, you can get, that's for sexual or genetic recombination, but you can get variety another way too. What's another way you could get variety? Not sexual recombination, or not recombination, but variety. Starts with an M. Mutation. Exactly. Through mutations. And because bacteria have a short lifespan and can replicate quickly in vast numbers, mutations can work for them because the best adapted organism usually is the one that's going to survive and reproduce further. They don't work as well for us. Okay, we talked about, you don't really need to memorize shapes or gram negative or gram positive, but how they act in their environment. We talked about some are obligate aerobes, they require obligate oxygen. Some are obligate anaerobes. They do not survive in the presence of oxygen. They can hide out as endospores. Some are autotrophs, some are heterotrophs. Some heterotrophs are even photoheterotrophs, where they use light to generate ATP, but they still need to consume carbon. Autotrophs, we talked about the two types. Phototrophs use light, and chemoautotrophs are ones like you would see in a hydrothermal vent that are using the breakdown of inorganic molecules to give them the energy in order to generate ATP and then reduction of CO2 into glucose. Yes, did I see your hand up?
Yeah, so just to make sure, we don't need to know about gram-positive and negative.
Whatever I put tiny, I don't know because I'm just talking off the top of my head.
Okay.
So I don't remember exactly what I put tiny. I thought about it when I did it, so, but I just know it in my head, so I'm just saying it.
Oh.
So tiny stuff, no. No tiny. We talked about uses of bacteria, things in food and food production. We talked about that they oftentimes are in symbiotic relationships with another organism. It could be beneficial in a case like mutualism and cyanobacteria in root nodules doing nitrogen fixation. It could be I don't care either way, commensalism. They could be taking up a space, which actually borders on the edge of mutualism for us, because by them living in our bodies, then something what? Bad isn't in that spot, okay? And then they could also be a very negative relationship for us if they're a parasite and they're making us sick. Then that's not working out so well for us. Several different diseases out there, chemical warfare. If I was going to put one in the Hall of Fame, though, I said it would be cyanobacteria. And I gave you a couple of reasons for that. Why? Yes?
They cause the oxygen revolution.
Oxygen revolution, they're at the bottom of the food chain, big time producers. What else? Has to do with the symbiotic relationship too, that we just went over. You know this one. Nitrogen fixation, yeah. Remember, most of the air going in and out of your lungs right this moment, most of this air, 78% of it is nitrogen. You can't do a damn thing with it, okay? We don't get stronger, we don't feel full, so we need another organism to get it in a form that plants can use, and then we can eat plants or we can eat animals who have eaten plants, and that's how we get nitrogen in our body. Who can do that for us? Cyanobacteria can help plants get it in their bodies. Okay, then last but not least, we talked about Archaea, and also another prokaryotic organism, and we know them as what? What's another name for Archaeas? Extremophiles, right? They live in these extreme environments where it's very salty or it's very hot or there's a low pH. And one thing to remember is that they can provide nitrite to oceans and water. And there's something else I was going to show you, but I can't remember what it is, so we won't worry about it. So what's this one right here? I'll let you answer it. And Archaea are more closely, even though they're prokaryotic, more closely related to what? Your eukaryotes. Good. So remember, when something says which is not true, then assume all of them are true, right? And just keep going, is this true? Is this true? Is this true? Yay! We don't have to do this one. Let's just talk about it. What's the first one? This one. Mutualism, commensalism, parasitism. How does this not an example of symbiosis? Yeah, they've got to be different species if it's symbiosis. And those are showing you the 3 domains, and that's the end. Any questions you want to ask me? Yes?
Well, on this quiz, will we start with showing questions from previous chapters?
No, this is not going to be a shadow quiz. That doesn't start till chapter 23 or 24. I thought first quiz out of the chute. What?
Isn't that the same quiz? 23 and 24?
Yeah, 23/24. Yeah, no Shadow Nation in this one.
Three chapters.
Yeah.
Protest.
All right, anything else? All right, Smarties. Make good choices. Have a piece of toast.